very important talk, and it's important that all of us should know how many of these drugs are tested, because many times it's like we should know how somebody is putting an effort to cook. If you don't look into their kitchen, then you assume that everything comes by magic. So drugs are always developed like this, whether you want to use a blinder on it, or you want to put an effort to understand this. And Tirumlesh is important because he's the first one of the first few who actually wanted to work on an animal model himself, hands-on work. Many clinicians would not want to do that, and he became so good at that that he's better than a lot of our lab scientists who actually work on the animals all the time. Interesting, Dr. Mohita. Small trivia. I mean, a lot of us here are dog lovers. Arka used to tell me when he worked on the gene therapy work with what Pooja said, initial work, he has a patent for the AAV vector on Stargardts. You actually have to work on dogs, because that's the end. Imagine you take a 2 3 big Labradors, they grow with you, they're in your lab for 4 5 years, right? And you create a model for them to go blind, means it's not blind, and then you inject it, and you have to euthanize the dog. You have to kill the dog yourself, and until unless you're able to do it, the lab will not take you. You can't say that I'm not going to do this on my lab. And those dogs you're grown with, they're not something you got from your pet shop. So it's science, or advancement of science is not as simple as we have understood it to be. Quickly, just go to quickly few slides. I know you have a long list of it, but
Actually, thank you, sir. Why we require animal model is because the same kind of disease you can see here that the amount of hypoxia and the amount of neovascularization does not match. In this slide, you can see although there are peripheral dropouts, you can see the neovascularization is more central. In this particular slide, you can see the capillary dropout is not too much, but still you see neovascularization. So there is no standardization when it comes to how patients present to us and what is the level of the disease. So before going into how we develop the model, we usually collect data. So before I joined NN, already NN was in this process where we had collected the aqueous and we analyzed many factors based on the cytometric bead array of the aqueous. Based on that, we narrowed down to few targets. So we already know that VEGF-A is already predominantly targeted and steroid kind of carpet bombs everything. But we hit on a soluble ICAM molecule because this was something which was present both in treatment naive patients, but it kept on increasing. And the good thing about ICAM is that ICAM is present right from the beginning. So these are the two guardians of retina, the endothelial tight junctions and the RPE tight junction. So whenever you stress these two cells, the first molecule that responds is ICAM, which means that this is the initiator, this is the one which is present during the disease process, and this probably is also present in the proliferative disease phase as well. So that is why we chose this particular molecule. And this is how we stressed the RPE. We took a cell culture line and we produced different kind of stresses and we examined the expression. So whenever we put them under stress, both hyperglycemic or hypoxic, they produce ICAM. So based on this, if you remember diabetic retinopathy, we said that whenever there is hyperglycemia, there is going to be leukostasis. That is one of the first thing to happen. Now, what produced that leukostasis within the intraluminal side is the expression of this ICAM. So that was the reason why we started modeling this, and we checked it before we went up. And many might ask a question that, oh, you were looking at aqueous. So this is a soluble protein that you're looking at. Does the RPE and the human choroid, does it have these ICAM? So you can see that in the donor eyes, we were able to in diabetic eyes, when we examined, the ICAM was active in all of them with the fluorescent antibody. So going to the animal model, so we know that SD rats is the standardized diabetic rat model. So we took the SD rats and we gave an intraperitoneal injection of STZ, which produced diabetes in a period of 12 weeks. So after 12 weeks, what we did was we gave anti-VEGF, which was ranibizumab, because that is the standard of care molecule. And we used our LFA1 antagonist, which tends to block the ICAM level. And what we did was, before we sacrificed the mice after 2 weeks, we put a Fitzy in its tailwind. Fitzy is like fluorescein that we give for patients, but in rats we use Fitzy. So this is what we saw. In the rats which were sacrificed without any diabetes, you can see that the vascular integrity is well maintained. Rats do not have macula. So any part of the quadrant would look very similar. So you can see that there is absolutely no leakage. But the moment we examined the STZ rats, which were the diabetic rat model, you can see that there is extensive leakage, presence of microorganisms, and all that. So if we add the ICAM to this, the leakage became more pronounced. You can see between that slide and that slide, that is more brighter because it's leaking from everywhere. So along with VEGF, ICAM definitely has a role which shows that it not only produces the disease, it also increases the severity of the disease. So our hypothesis kind of became more and more stronger. Now, when we add the antagonist, you can see the beauty of it that from there it came there, and it was as good as the anti-VEGF. In fact, if you look between the anti-VEGF and the STZ, I mean, and the LFA1 antagonist, which I put side by side, this can even produce stability of the endothelial tight junctions as well. So we were thinking that this is one molecule which probably would influence the treatment as it goes further. Now, to make the hypothesis a little more stronger, what we did was we took the wild type rats, which were not transgenic mice. We did not give them diabetes this time. What we did was we used a vector and we overexpressed the ICAM molecule. And we did not feed it anything differently, but you can see between the control molecule and the ICAM expression, overexpression molecule, you can see there is definitive presence of microorganisms which is already there in that part. If I can show there, right? So this makes more and more sense that ICAM definitely has a role, and this particular molecule can play a role in future. So the second part of it is AMD. India is a very young country. That's why we became so popular with the US that they were outsourcing our people to there. Very soon, we are also going to end up in an AMD epidemic. So that is one of my very interested topic which I'm working on. And Dr. Sina is our collaborator at Pittsburgh University, because India does not allow studies on monkeys and chimps. So the NHP studies are done in Seri by Dr. Bharti. So you know that AMD has about 200 different genes. But the beauty of it is, if you knock off any of those 200 genes, you will never be able to produce a AMD in animals, which means that every single gene that has been reported till date is an associative gene at best. It's not a causative gene, right? But Dr. Sinha's lab stumbled on this particular gene, which I'm not supposed to reveal, so I have not revealed it. But when they produce a knockout of that gene, the beauty is that they produce the first model which actually can show drusense in mice. I mean, drusense in mice is something which is absolutely unheard of. And when we were working on it, we found a transgenic sequence. So NN is providing the transgene, which is being produced in India at our lab, and we are also producing the vector here. So they are providing the expertise of the gene sequence which is supposed to be altered. So that's how we are working, and we are already finished the rodent trial, and we are in the non-human primate model. So this is one drug which will very soon come, and this is something where you can actually start giving in patients at some point of time, probably a span of 2 to 3 years. And current existing molecules talk about gene therapy where you have to do a vitrectomy, induce a retinal detachment, and give a subretinal injection. So if a AMD patient comes, you're going to tell them that I'm going to detach your retina and give. That patient probably might not really agree. So the challenge that the university gave us was, can you give us a slightly better option? So at NN, we have developed a patented needle which can allow the molecule to be delivered into the suprachoroidal space without any retinal detachment. So ideally speaking, just like how we give an intravitreal injection, this particular drug can be delivered at office, and we use the same needle. I'm not showing it because of patent issues again. So we delivered the drug, and you can see in baseline first and second injection, whatever we have seen, it has not produced any toxic effects.
And now, I think if I'm right, you are moving this from
Primates to
small
humans.
primates to chimpanzees.
This is in chimps already, sir.
Chimps. So chimps, we don't have an animal model of chimps in
Yeah. So we are
India.
aging them, and then we are.
So they go, yeah. So we'll have to work with Singapore Eye Institute for work on a bigger primate, because that's the only one which is closest to.
The first thing they wanted to know is it toxic? So we followed them up. We did an OCT. We did in the post-injection fluorescein, and there was no toxicity at all.
I think
And
that should be
this is the last one that I would want to show. That is it effective? You can definitely see that the gene that we gave was able to produce presence of the RP, which was almost dying. It did tend to revive it in all of the
that
of
the green one
the
is.
green one, the hyperfluorescent one, is proof that this gene gets integrated and actually alters.
And this is on what animal?
This is the chimp only.
Okay. Any questions by anybody? This is the last one. So basically, I think, like the sir mentioned, there is a lot of work we can work together with collaboration. Dr. Moita has shown this, and companies like Cipla have to look beyond just organizing meetings. You'll have to look beyond, like you have done work with Sharon. I think ultimately it's going to help you. We have Dr. Suzuka Ko from visiting from Japan. You want to add anything? She is a cornea surgeon in Kyoto University and does a lot of work on biomechanics. Has been her area of work. So you have an animal house lab in University of Kyoto. Kyoto is known for more of optics. Osaka, yes. Anybody has anything? If nobody has any questions, thank you all. I thank Cipla for this wonderful event. And Nitesh, you want to say anything? Absolutely. Doctor.
Continuing with this kind of good work, whether it is
So-
form of rare disease or it is in a form of a disease which is very well known to us. And the way, yeah, the way the sir was talking about, I think Cipla was the first organization which developed depriprone for thalassemia patients. And this is an iron chelator, which is thoroughly, it's a service product for the pediatric population of the patients. And there are a good number of drugs which we have introduced only for this kind of population where nobody else is there. And we'll talk about pandemic. We're talking about COVID. COVID was, I think, such pandemic where Cipla has come out full-fledgedly with all kind of drugs. Whether you talk about remdesivir, whether you talk about Actimera, whether you talk about collaboration with everybody. So we are always there to help. And for us, we also say that the patient comes first, and with you, we try to reach to the patient. That's our objective. Thank you, everyone.
It's just a suggestion. So now you can do large animal studies in India, and it will take only 1 month to get it clearer. Okay? Not me, to the large animal committee, not me. I am not responsible, but this is the latest.
Thank you, sir. We didn't know.